A nerve impulse transmitted from one neuron to another neuron through junctions called synapses. Called synapses. Synapses. See here. Next again. Like this. Nerve impulse transmitted from one neuron to another neuron through junctions. These are called junctions. These junctions are called synapses. Synapses. This is the definition, one mark definition. Nerve impulse transmit from one neuron to another neuron through junctions. From one neuron to another neuron through junctions. These junctions are called synapses. So, an action potential, already we discussed that. An action potential is transmitted. An action potential passing down and reaches to end region. End and its branches. An action potential passing down along the axon and it reaches to the end and its branches. These branches forms junctions with another dendrites of neurons. These are dendrites of neurons, muscles, and with secretory glands in the muscles. That is the second step. See, okay. So action potential passing down along the axon and reaches to terminal surfaces. Passing down along the axons and it reaches to the terminal and it's all the branches. Branches means what? Here branches are there. Branches. Synaptic bulb reaches to the region and it is connected to all its branches. These branches forms junctions with another dendrites of neuron. Forms junctions with dendrites of another neuron, muscles and secretory glands in the muscles. That is the first point of the transmission of impulses. Next here, synaptic cleft. Synaptic cleft. Synaptic. Synaptic cleft. Synaptic cleft. It is an intercellular space. It is an intercellular fluid-filled space that separates axon terminal and another target cells. That is called synaptic cleft. You will not understand. This is if I will draw a diagram, through that you can get better understanding. Synaptic node. Next, here, this is what? Dendrites of another neuron. In between these two, this is called presynaptic membrane. Presynaptic membrane. It is called postsynaptic membrane. Presynaptic membrane and postsynaptic membrane. In between these two, gap is there. This gap is called synaptic cleft. Synaptic cleft. synaptic cleft. Here that only I am saying. It is synaptic cleft. It is an intercellular fluid filled space. This separates axon terminal, that is presynaptic membrane, and another target cell. It is another target cell, that is postsynaptic membrane, that is called synaptic cleft. Synaptic cleft. Okay, that's it. Here, types of synapses. Types of synapses. Depending upon the nature of transmission of information, synapses are two types. It could be of two types. One is electrical synapses. Another one is chemical synapses. See here, depending upon the nature of the transfer of information, transmission of information, synapses could be of two types: electrical synapses and chemical synapses. Electrical synapses and chemical synapses. First of all, let us see electrical synapses. These are special type of synapses. These are very, very special type of synapses. These synapses very rapid. These are rapid for signal transmission. For rapid signal transmission. Rapid signal transmission. That is first point. These are very, very special for rapid signal transmission. Okay, electrical synapses. So the information is transmitted rapidly. So that's why these are rapid signal transmissions. Signal transmissions. Second one is, see here, electrical synapses. Through this rapidly transmits signals from one neuron to another without any delay. So there is no gap. Electrical synapses. There is a small gap, that is synaptic cleft. That is the synaptic cleft is narrow. It is about 0.2 nanometers. See here. Actually, generally I have drawn. If you will see, this is in electrical synapses. Very small. It is very narrow, small. That is about 0.2 nanometers. The length, the gap, very small, narrow. That is about 0.2 nanometers. There is no signal delay in this one. Signal delay does not happen. There is no delay. There is no synaptic delay in electrical synapses. That means what? In electrical synapses, signal delay does not take place. The signals are transferred instantaneously. From presynaptic neuron to postsynaptic neuron. From one axon terminal to another target neuron. Without any delay, signals are transmitted instantly from one cell to another cell. Instantaneously means immediately, less than millisecond. Okay, so in such a way it will be transmitted. Generally, so this kind of signals occurs in human retinal ganglion cells. Human retinal ganglion cells. Signal transmission occurs through electrical impulses. Electrical synapses in bidirectional. Bidirectional means what? From our eye to brain, brain to eye. Bidirectional electrical synapses in human retinal is bidirectional. Human retinal ganglion, the masses of nerve cells. In them, electrical synapses are bidirectional from outgoing to from here to from retinal to brain, brain to retinal. That is the information is called electrical synapses. Ultimately, 2 points you have to remember. There is no delay in electrical synapses. It could be rapid for transmission of signals from one cell to another cell. That is called electrical synapses. Okay? See. Chemical synapses. These are commonest kind of synapses. Generally, commonly in our body, remaining body parts, synapses are transmitted through chemical synapses. Okay, so these are commonest kind of synapses. Neurons transmit information from one neuron to another neuron through chemical transmitters. Chemical transmitters. Those are called neurotransmitters. Those are called neurotransmitters. The neurotransmitters are excitatory neurotransmitters, inhibitory neurotransmitters. Here neurotransmitters are again 2 types. Excitatory neurotransmitters and inhibitory neurotransmitters. Both are important to our body. Excitatory neurotransmitters, inhibitory neurotransmitters. Excitatory neurotransmitters, that is acetylcholine, glutamate, epinephrine, and norepinephrine. These are excitatory neurotransmitters. Acetylcholine, glutamate, epinephrine, norepinephrine. These are excitatory neurotransmitters. Next one is inhibitory neurotransmitters. Gamma aminobutyric acid. G stands for gamma aminobutyric acid. Glycine, dopamine, serotonin. These are all inhibitory neurotransmitters. So these will inhibit the transmission. Neurotransmitters inhibit the nerve impulses. These are excite the nerve impulses. Okay, so the chemical synapses, these are commonest kind of synapses. These synapses passes through the neurons with the help of chemical transmitters. Those chemical transmitters are called neurotransmitters. These neurotransmitters are secreted by secretory cells present at the synaptic knob. Already discuss that. Okay, that's all. This next continuation. See here, that is first statement, second statement. Here, the synaptic cleft, the gap between the presynaptic membrane, post-synaptic membrane. The gap between pre and post-synaptic membrane, that is synaptic cleft. Synaptic cleft. Synaptic cleft. This is called synaptic cleft. Is wider, and is about 10 to 20 nanometers. The gap at chemical synapses. The nerves which shows chemical synapses. In them, the gap between the two terminals. It is axon terminal and it is another dendrons of another neuron. This is called synaptic cleft. Its gap is at chemical synapses is more wider. That is about 10 to 20 nanometers. 20 nanometers. Okay. Chemical change at the synapses. Chemical change at synapses. Chemical change at synapse. First of all, and action potential. An action potential. Action potential means nerve impulse reaching to the synaptic knob. What is synaptic knob? As you know that it. At the end of the axon is called nerve terminals. At the end of nerve terminals, say bulb-like structures are called synaptic knob. So these are myelin sheaths. So an action potential reaching to the synaptic knob. An action potential reaching to the synaptic knob. So an action potential stimulates. An action potential stimulates opening of the calcium ion gated channels. So those action potential we cannot see with our naked eye, but it will reach. When it will reach it to here, those action potential stimulates, induces the opening of the voltage gated calcium ion channels. Here, voltage gated calcium ion channels will be there. Voltage gated calcium ion channels will be there. Voltage gated calcium ion channels will be there. So here, voltage gated channels. Voltage gated calcium ion channels. It stimulates the opening of the voltage gated calcium ion channels. So when calcium ion channels are open, these calcium ions are diffused into the cytoplasm of synaptic knob. These calcium ions are diffused into the cytoplasm of the synaptic knob. The cytoplasm of the synaptic knob contains numerous mitochondria, which is responsible for the production of ATP. These are all our energy expenditure processes. That energy who will provide? Mitochondria. That is ATP. Okay. So listen. Action potential stimulates the presynaptic membrane to open the voltage gated channels of calcium ion. So when calcium ion gated channels are open, this calcium calcium ions are diffused into the cytoplasm of the synaptic knob. In the synaptic knob, cytoplasm of the synaptic knob, here it contains numerous mitochondria. Those mitochondria are responsible for the production of ATP. Okay. Next, third statement. See, these calcium ions moves the synaptic vesicles present in the synaptic knob. These calcium ions is here number of synaptic vesicles. Synaptic vesicles These calcium ions move the synaptic vesicles rapidly in the cytoplasm of the synaptic knob and fuse them with the presynaptic membrane. As synaptic vesicles are fuses with the presynaptic membrane. Fuse the presynaptic membrane. After fuse, these synaptic vesicles are ruptured and releases neurotransmitters by exocytosis. Bulk of neurotransmitters releasing is called exocytosis. Exocytosis means bulk. So calcium ions are allow the movement of synaptic vesicles and fuses with the it is fused with the presynaptic membrane. As the synaptic vesicles fuse the membranes, rupture them and releases neurotransmitters into the synaptic cleft by exocytosis. When the neurotransmitters are released into the exocytosis, immediately, without any time delay, they are transferred into the post-synaptic membrane. It takes about a millisecond. When released from this synaptic cleft, immediately they will bound to the post-synaptic membrane. It takes about a millisecond. Here, one more very important information we have to discuss. See, this is post-synaptic membrane. Post-synaptic membrane means what? Dendrites. It is the dendrites of other neuron or muscles or target cell. Here what happened? When these neurotransmitters are released, before binding, here what is there? Potassium ion, sodium ion channels are present. Potassium ion, sodium ion channels are present in dendron of another neuron that is on post synaptic membrane. So along with these potassium protein molecules. Those protein molecules are called receptors. These are also called chemoreceptors. Chemoreceptors. So these neurotransmitters are attached to the chemotransmitters with the help of potassium ion and sodium ion channels. Here it will generate the second action potential. Second action potential. Neurotransmitters are attached to the postsynaptic membrane. With the help of chemoreceptors and potassium ion, sodium ion gated channels, here second action potential are generated. The second action potential is continued. This is called chemical synapses. Chemical synapses. Chemical synapses. There are different neurons. There are more than 30 different types of neurotransmitters have been identified so far in human body. 30 different types of neurotransmitters have been identified. So with this topic is finished. Types of neurotransmitters. Types of neurotransmitters. There are 30 different types of neurotransmitters have been discovered so far. Those 30 neurotransmitters have been classified into 4 types based on their chemical structure. First one is acetylcholine. Acetylcholine. Second one is biogenic amines. These are also called adenine. Amino acids. Neuropeptides. These are called endorphin. Endorphins. Different neurons bring different types of neurotransmitters that may be excitatory or inhibitory. Excitatory neurotransmitters I said, inhibitory neurotransmitters I said. In such a way, 30 different types of neurotransmitters have been discovered so far in human beings. Those are acetylcholine. Depending upon chemical structures, they have classified into 4 types. Acetylcholine. Biogenic amides, these are also called adenine. Amino acids, neuropeptides, these are also called endorphins. Okay, these are the neurotransmitters information. With this, transmission of impulses is